Hi, thanks for having me, Roger. My name's Aaron Edwards, and I'm the CEO and co-founder of Kiragen Bio. And we're on a mission to engineer multiplex gene-edited cell therapies that can overcome the suppression from solid tumor microenvironments. So really quickly to give you some background, CAR T cells and other types of engineered immune cell therapies have really led to transformative benefits in the hematologic or blood cancer space, really transforming that entire clinical area. But unfortunately, that efficacy and transformation has not seen its way into solid tumors for various reasons. I'm listing here on the left three of the main key barriers to why solid tumors have not worked in the past. It's antigen heterogeneity. So what can these cell therapies, specifically CAR T in the context for Kiragen, target on the tumor cell? It's a little bit more complicated for solid tumors. Impaired trafficking has always been a question. If you're giving these cells via IV infusion, can the cells actually get to the solid tumor site? And then once the T cell or the CAR T or immune cell therapy gets to that solid tumor site, it then encounters this tumor microenvironment, which I'll talk about in a bit, that also needs to be addressed to make sure that these cell therapies function. I'm showing a really recent example of this in glioblastoma, where researchers at UPenn and MGH has had similar data showing that if you address two out of the three of these barriers, you could get efficacy in CAR T in solid tumors for one of the first times ever seen. Unfortunately, that efficacy is not durable. So you can see in the first panel, day -1, so before treatment, day one after treatment, you're seeing both of these sites regress via MRI. But unfortunately, by day 33, almost all of those patients do relapse with their tumors once again. The root cause for this is that tumor microenvironment that I was mentioning earlier. There are not just tumor cells that are spitting out, say, offenses and defenses to turn off these cell therapies. There are also other immune cell types that are coming in. So once a cell therapy can get into that solid tumor, it starts doing its job. But part of that job is releasing chemicals that can attract these other immune cells that are all meant originally to suppress other cell types or keep a regulatory function in place. So there are many different pathways that can really be used to turn off all of these cell therapies, and we've bucketed them into basic categories of immunological, so these immune cell types, or PD-1 and CTLA-4 or inhibitory receptors, biochemical, so they can spit out these proteins that turn off the cells, and then really just a toxic metabolic environment. So low nutrients for these cells to really function. And while that might be overwhelming for the majority of people to know that there are over 50 pathways that you could target, and how would you ever choose? For a gene editing and a combinatorial gene editing company like we are, it gives us a really broad opportunity to actually enable some combinatorial strategies for our products. So there's a lot on this slide, but really focus on the cartoon in the middle. So our solution falls into a different bucket than other categories today. So a lot of companies, and you see the logos here down on the bottom left, they really relied on supercharging. So making that T cell or cell therapy more potent so that when it reaches this suppressive environment that's around these solid tumors, each T cell packs more bang for its buck and can break through that solid tumor suppressive barrier. That has some interesting characteristics, but it does unfortunately pose some neurotoxicity concerns for really sensitive indications, especially in the brain. And our guarding strategy tries to flip the script on all of this and say, if we can now multiplex edit instead of relying on these nucleases that are like scissors to cut up the genome, we can now focus on actually solving the problem directly instead of sort of redirecting function that these other supercharging strategies have. So going back to the suppressive solid tumor microenvironment, the T cell or those cell therapies we have are sensing those suppressive factors from receptors on the cell surface. So that's what's shown here in the cartoon. On the right, we have the ability to knock out not just one receptor, but many different receptors that are seeing those suppressive factors in the first place that essentially protect the cell from suppression and allow that CAR T to function normally. So if our goal is to translate what we've already seen in blood cancers to solid tumors, this allows that CAR molecule, which we know to work clinically and in humans, it allows it to function hopefully long enough to enable really lasting, durable function in these patients, especially for glioblastoma patients, which is our lead indication. We've created a problem for ourselves by being able to multiplex edit so many pathways at once, is to be able to rationalize those decisions. And I'm not going to go into this in much detail, but we do have a machine learning model that can really rationalize and navigate these billions of editing combinations. If I told you that there were over 50 tumor microenvironment components that we could choose from, and our hypothesis at Kiragen is that you need to overcome multiple of those. We're saying five plus to really have durable efficacy of, against these suppressive factors. That's over 10 billion permutations of combinations that there's no way we would be able to get through empirically or in a cost-effective way. So our machine learning model, along with wet lab validation, can help select the most ideal combinations to test that are really optimized for each of these solid tumor indications and allow these cell therapies to really function best in each environment. We do have in vivo validation for not only our lead program in glioblastoma on the left, but also a non-small cell lung cancer indication on the right. And really, what these two graphs are showing is that in white is untransduced or just really showing the tumor alone growing, but that we can layer in more than one guard at a time, and each of these can lead to more benefits in these mouse models. So in the black, you're showing what's an unmodified CAR T cell. It can, if for glioblastoma, it can actually control, just like I showed you in the MRI in the first slide, but unfortunately does relapse since it's not able to hold a durable control. Red is knocking out one of these pathways. So the one that's listed here is TGF beta, which is a really common suppressive factor in the tumor microenvironment. And we're showing in some indications, like the lung cancer on the right, you might get some benefit. But for something like glioblastoma on the left, you only get a little bit added benefit. And it really requires multiple guards to be in place to really have durable control over these really suppressive tumors. And this is really showing that our approach is portable across solid tumor indications and is really agnostic to the sort of type of binder or cell type that you're really using. Although CAR T is where we're really putting all of our efforts right now on the Kiragen front. So we are now working with the team at UPenn to bring a next generation CAR T back into the clinic. It is off the shelf. It has, sort of some safety advantages over these potency plays, and it is suppression resistant by having five knockouts in a single product that directly address those key relapse drivers I was showing in the first slide. It is best in class, first in class. These glioblastoma patients do not have other treatment options. And so if this works from a business perspective, what is best in class is first in class and vice versa. And it does have some advantages over these non-cell-based biologics, which are limited on what they target, but also rely on the host immunity to actually drive durable function when our cell therapies are doing the direct activity against these tumors. We have a scalable manufacturing process, so we can scale our dosing down to 40,000 per dose, which is way less, probably a tenth less than the current CAR Ts that are on the market that are patient-derived. We actually use an allogeneic process, so these are healthy donor-derived products. This eliminates manufacturing delays, lowers COGS, and really increases access. And going back into the clinic with UPenn through an investigator-initiated trial really fast tracks all of this at a fifth of the cost, really on time. So we could get into the clinic today with an IND around 2027. We have different pathways in terms of funding to really get there, that are starting from this entry point, which is 2.5 million, but we have really carefully mapped out each check size that can go all the way through a clinical trial, depending on some of these investors that have larger checks to write and might want to, push Kiragen forward within a single fundraising round and a single check. So, happy to let you look at these afterwards if you're interested. And it really maps out asset, platform, how far pharma validation gets us, and then really fundraising readiness to a Series A raise. For quickly on the market, GBM is a $5 billion market, but that also correlates to a pretty well, it would be a complete market share, unfortunately, because these patients don't have any other treatment options available. But we do have long-term plans to expand into not just solid tumors, which represents about a $300 billion market, but also liquid tumors as well, where we're already talking with some pharma companies to really improve their current blood cancer franchise. And quickly, these are just really three phases to get us to where we want to go. This unlock phase, which shows activity among our main hypothesis here around tumor microenvironment suppression blockade, advancing into other programs such as brain metastases and CAR T outside of the brain, and then finally expanding into other different cell types because our product and our sort of approach is very modular in nature. This is a great team that we've assembled. Ryan Murray and I have known each other for about 10 or 12 years now. Worked together at both Bluebird Bio and Beam Therapeutics, where I led the in vitro biology for Beam 201, which is their now clinical CAR T program. Tessa Green leads all of our machine learning. Got her PhD recently from Harvard Medical School, and Mark Fick leads all of our platform and manufacturing activities. Also worked with us both at Beam and Bluebird, and most recently at Tome. A great set of advisors, and we have a lot of support from the Blavatnik Family Foundation and the Termeer Foundation as well, to really give us all the support we can to hopefully give us the best foot forward, as this is a hard market to be a startup right now, but we have conviction that we will make it through, and we're really excited to have anyone that might want to support our mission, either with belief capitals that we so to speak, or financial means, or really just being able to share our story. We really do believe that this possibility of blocking the suppression will unlock this full potential for cell therapies that can transform the lives of patients with these intractable cancers. I'm really thankful to have the opportunity to share a story today. So thanks a lot, Roger.